everyone. So this is Shruti Ahuja, and in this video we are going to start a new series of histology in 3 to 5 minutes. So I will be covering one topic or at least one slide in one video. I will be telling you about all the important PYQ, the diagram, the slides and important identification points which you can write in the OSPI exam as well. So this first video is very basic. These slides won't be coming, and I will just be telling you how we can start learning and what are the basic points which we must know if we want to learn histology in a very conceptual manner. So first of all, we know the simple epithelium, right? Simple means single layered for this context. So here we can divide it and classify it according to the shape of the cells, right? So shape of the cells can be flat, it can be square, or it can be rectangle, that is the columnar, and it can be multi-layered, okay, polygonal, or we can say not having some specific shape, polygonal, different shapes, and multi-layered, looking like multi-layered, right? So from basic shapes you can understand it. So say, if it is flat, then it is simple squamous, right? So here it is simple squamous layer of flattened cells with flattened nucleus. So you need to note this point that if you draw this circular nucleus here in simple squamous, then your marks will be deducted, and most of the times we do this mistake and we don't know why our marks are cut in histology diagrams specifically. So here the examples we know: endothelium of blood vessels, mesothelium, lining of alveoli of lungs, etc. Next, simple cuboidal. Cuboidal we know it is like square kind of thing, right? So the breadth and the height is equal. Here breadth and height are equal, and the nucleus are like central and round. See, I have drawn it in center and round. Next is simple columnar. Columnar like a column. We know column is like straight verticals. So it is like a columnar. You can see it here as well. And the nuclei are on base and they are oval. So this way you can understand, and if you will draw it in the same way, then only it will make sense, and this will also help you in identification of the slides when you will go for the OSPI and the grand vivas. So example of simple columnar are the mucosal lining of the stomach and gallbladder. Just remember the GIT, columnar. Okay, it is a thumb rule. Next is simple cuboidal. The examples: cuboidal. If you will go to any duct of the body, in 99% you will find the cuboidal, right? So the collecting ducts of the kidney, the follicular cells of thyroid. In some ducts there is exception, it does exist, but remember mainly the ducts of the body. Next is pseudo stratified. Pseudo means false, stratified means multi-level, right? Multi-layered. So it is not multi-leveled, but it is looking like it. How it is possible? If I place one brick here or one brick there and one brick here, it will look like from a distance that the thing is multi-layered, right? This way I can say that if this is my columnar epithelium and I have placed one nucleus basal, one is at top and one is at center, then it will look like at multi pseudo stratified, right? Then we can say that it is many layered. The example of the epithelium is respiratory epithelium, and respiratory epithelium you can see it here. This is the respiratory epithelium. Here this one, this is looking like this, you can see this is looking like single layer and this is looking like multi-layer. So this difference you can see of the epitheliums. This is pseudostratified and this is we can see the simple squamous of the alveoli. So this was about this. These are the diagrams given in the manual as well. You can compare these diagrams with my diagrams as well. This will help you to draw in exam as well. Okay, so this was all about the simple epithelium. In the next video we are going to talk about the stratified epithelium. So stay tuned, and if you like this video then do like it and share it with your friends, because sharing is caring.